Okay. Well, we want to welcome all of you to our PolarConnect. We're excited to have DJ Kast and two researchers, Dr. Crump and Dr. Kling, with us today. We're going to be learning all about their expedition and their research that is being conducted up at Toolik Field Station in Alaska. And today is July 7th, 2016. Before we get started and hear from the team, there's a few things we need you to know about using this platform. So if you've joined us by phone, again, I need to make sure that you mute your phone, *6 to mute, *6 to unmute. If you're using a microphone today to ask your questions, I need you to click on the little microphone above the window. There's a little microphone way up there, and turn it off. Click it once to turn it off, and then later on we'll let you click it on again to turn it on and ask questions. During the presentation, slides should be showing up here in the center. There's a list of participants on the far left. We also have a webcam. DJ's connection is not great, but she might come on at the end of the presentation to say hello and respond to questions. There's a chat feature in the bottom left. You can type in where you're from, ask your questions there, and post any comments and things like that as we go along. There's also a couple of other features with Adobe Connect. There's the PDF is also available online if you want to download it, and some links that kind of talk about the program and links to things that you might be of interest in. So while we talk about PolarTREC here in a moment, if you don't mind doing so, please let us know where you're from, and if you have participants that are sitting with you, who they are. That kind of just let us know where everybody's coming from. And real quickly, I just want to go over what the PolarTREC program is and why DJ is part of this program. The PolarTREC program stands for Teachers and Researchers Exploring and Collaborating. It's administered by a nonprofit that Judy and I both work for, the Arctic Research Consortium of the United States. We are primarily based in Fairbanks, Alaska, and have an office in Washington, D.C., and we do a lot of work with the Arctic research community in helping them reach out and share what they do, share with other researchers, share their research with communities, and we do education programs like PolarTREC. PolarTREC has been around since 2004, and we've placed over 150 teachers from around the United States with Arctic scientists and Antarctic scientists like Byron and George. And we are really proud of our program. It's an opportunity for teachers like DJ to get hands-on field experience and bring it back to the classroom. So that's why she is part of our program, and part of the things that we have teachers do is reach out to the public about the science, and the PolarConnect event is one of those opportunities. So for your questions today, as we go along, you can type your question in the chat box. At the end of the presentation, you can raise your hand and we'll call on you, and you can open up your mic or unmute your phone and ask your question. We'll go over those instructions again at the end. But in the meantime, as we go along, please type them in the chat box. Like I said, we do have both researchers here today, and they'll help also field questions. So with that, I'm going to turn it over to DJ if she's ready.
Yeah, I'm definitely ready.
All right.
So my name is DJ Kast, and I am the STEM Programs Manager for the University of Southern California. I have a joint appointment with the USC Joint Educational Project and the USC Neighborhood Academic Initiative, and altogether it's essentially a K-12 or kindergarten through 12th grade STEM program for about 3,000 students that are low income and underrepresented minority students. And I have been working primarily with Dr. Crump and getting some wonderful feedback also from Dr. Kling. Would you two like to introduce yourselves?
This is Byron Crump. I'm calling in from Oregon State University. Just got back from the Arctic this past weekend after spending a couple weeks up there with DJ.
And this is George Kling. I'm calling in from the University of Michigan. Byron and DJ and I spent, well, about 2 weeks up in Toolik together before Byron and I left. So we had a chance to talk about science and explore what's going on up there.
Wonderful. Thank you, Dr. Kling and Dr. Crump. Next slide. So as all of you might probably be aware, our world is changing, and the carbon dioxide levels in our atmosphere are majorly increasing. And this increase has dire consequences for humans all over the world. For places like where my students are, there's going to be extreme drought and heat. It was 115 last week. I know that's weather, not climate, but they're not liking it. We have sea level rise and floods, warmer global temperatures, and extreme weather, and all of those also have more consequences that will further the pattern. Next slide. And the Arctic in particular is warming faster than the rest of the planet because greenhouse gases don't always want to go and spread evenly across the entire planet. And so on average, as you can see by this picture, the land and ocean temperatures are increasing all over the planet, but the Arctic is particularly vulnerable, and it's warming faster than everywhere else. Next slide. And so there's many things that make the Arctic unique, and one in particular that I've been learning a whole bunch about up here is what's called permafrost. And permafrost is essentially frozen soil. So you can see that on both in the picture that my colleague Jason is standing on, you see the top layer, which is the active layer, and then a layer of frozen soil underneath. And a lot of roots can't penetrate further than that, but there's been like sometimes thousands or sometimes millions of years of frozen decomposition matter that lives right in there. And as the planet gets warmer, this also slowly starts to thaw, which is definitely not good for all of us humans here on the planet because it is a giant source of carbon. And it's organic carbon, and this carbon is going to be called soil carbon through the rest of this presentation. And that carbon is stuck in the permafrost as it's frozen. But what do you think is going to happen to the permafrost as our Earth gets warmer? Next slide. So right now I am calling in, yes, it will thaw. Thank you, Anne and Kara. And it will thaw and more carbon will be released into the atmosphere. And so right now I've been working with a whole bunch of different researchers up here, but primarily with Dr. Crump. And I'm at the Toolik Field Station, which is the little star at the northern part of Alaska in the dark purpley range. And the reason that Toolik is so unique, not only for being, an amazing field station up here in Alaska, but it also represents what change is happening in the Arctic, what change is happening in the Arctic throughout all of the Arctic, so within the Arctic Circle, not just what's happening here in Alaska. It's very representative of what's going on everywhere, and the researchers here are trying to predict how the Arctic ecosystems are going to react to climate change. And in this particular picture, it's the dark purple is where there is continuous permafrost throughout a particular area. Oh, thank you for the arrow. And this permafrost, these soils contain tons and tons of carbons. And if all of it were to thaw and be processed, we would double the amount of carbon dioxide currently in our atmosphere. So not good. Next slide. And what's worse is that this permafrost thawing has a positive feedback loop. And so as much as positive sounds good, it's not a good thing. The thawing of permafrost allows more soil carbon to be turned into carbon dioxide, which is then released into the atmosphere, which then raises temperatures and just continuously moves this forward. It causes more warming. It's not actually a positive thing. Next slide. So, and you can write it in the chat. Who do you think is causing this release of carbon dioxide from the permafrost? Anybody? Bueller? What do you guys think is causing the release of carbon dioxide from the permafrost into the atmosphere?
You're typing, they're thinking.
Well, it is thawing and decomposing, but who's the perpetrators in this particular scene? Who would do this? Who likes to decompose? Well, Dr. Kling has it all the way from the East Coast. It is all about the microbes and the bacteria. And so if you're looking at this visual right here, the permafrost is essentially a freezer filled with bacteria food. And as the temperature gets warmer, you take your food out of the freezer, you leave it out for a while, and that food starts to thaw. And those microbes, they love that thawed food. Permafrost is a whole bunch of, frozen decomposition matter, so they can just continue to decompose it. And they eat all that thawed food, and as they eat it, they let out carbon dioxide, just like us. Next slide. And so working with Dr. Crump, the focus has definitely been microbes, and that's because microbes are everywhere. Microbes, they eat soil carbon, and so and what's in the permafrost, and they breathe out carbon dioxide. And this rate of soil carbon, being consumed and carbon dioxide being breathed out depends on which microbes are where. So when you think of that, every environment has its own distinct bacteria that define a particular habitat. There's soil bacteria, there's lake bacteria, we have our own microbiomes or, defined bacteria that belong to us. And the microbes actually change, and the types of microbes in a certain area change depending on what's happening in the environment. And so that's where a lot of the research question that Dr. Crump's lab is focusing on come in. Which are the bacteria that are consuming the most carbon dioxide, and where are they doing that? Because bacteria, they're kind of like us. They have different types of eaters. They have people that are vegan. They have bacteria that are vegan. They have some that eat meat and potatoes and no vegetables at all. And we have some that only eat meat. And so specifically what we're looking at is the types of microbes that are consuming carbon and trying to quantify how much carbon dioxide is being given off by a particular species. Next slide. So you may be wondering, what actually is a microbe? And so in general, a microbe is an organism that's too small to be seen by the naked eye. And microbes are kind of an all-encompassing term, and the primary focus of what we're specifically looking at would be bacteria. And bacteria are single-celled organisms that will thrive in diverse environments. And so Dr. Crump's research team is particularly looking at how microbes impact the Arctic ecosystems and how they are essentially impacting climate change. Next slide. Oh, go back. And so microbes are very, very small, but they play a huge role in warming the earth and carbon dioxide production, since when they produce this carbon dioxide, that carbon dioxide can spread around the entire planet in just a few days. And they are decently small. When we have about a milliliter of water, there's over 1 million bacteria in that milliliter of water. So imagine we're looking at lakes and streams, and that's a lot of bacteria. And they're very, very small. They're about 100 times smaller than a wisp of a human hair. So small. Next slide. And so this is where I come in. I'm trying to translate some of the work that Dr. Crump's lab is doing. And specifically what we've been looking at is essentially an Arctic microbe crime scene. We're trying to figure out who exactly is there and what are they doing. And so, these microbes are converting the soil carbon we've been talking about into carbon dioxide, but different habitats have their own unique fingerprints through the lens of the different types of microbes that are there. So streams, lakes, and soils they're very different, but they are specifically defined essentially by a fingerprint of that particular community, and we define those communities through their DNA. And for the rest of the presentations, when we talk about lake, stream, and soil bacteria, lake bacteria is going to be identified by a pink fingerprint, the stream bacteria is going to be purple, and the soil bacteria is going to be green. But a lot of these habitats, especially in the Arctic, are all connected, and Dr. Crump's team is trying to see how these communities interact as they are washed down the watershed. Next slide. And so this was actually a wonderful paper or figure from a paper in Dr. Crump and Kling's from their lab. And essentially what we're studying is called microbiogeography, so how microbe populations change across the landscape. And as microbes travel down the watershed, they go through various habitats that have different streams, and you'll find a combination of them ending up in the lake. So in this particular figure, you have different types of environment. You have tussocks, you have willow, you have wet sedge, you have your soil bacteria starting on the left-hand side. You have streams kind of moving the soil bacteria through, slowly becoming stream bacteria, and then when they all meet together in the lake, you have both the soil bacteria, you have the stream bacteria, and you have the lake bacteria all in the same place. And primarily, what's most important is that bacteria in the soil are the ones that turn soil carbon into CO2, primarily. And once they get into lakes and streams, they do continue to do that, but there's different environmental changes. Like the temperature is different in soil versus stream versus lake. So is the pH. So is the water chemistry. And so the major question is, can these lake bacteria continue doing what the soil bacteria are doing? Is the lake bacteria also converting that soil carbon into carbon dioxide? Next slide. And so there are a whole bunch of different research slides research sites, not slides. I'm looking at slides. Research sites that focus on this. And this is a picture of what we're looking at right now. This is the I-series, which is a series of 11 lakes and streams that are all connected to each other and end up in Toolik Lake. Next slide. So as you can see here, if you're looking at this particular picture, each of the little black divots there are lakes, and then the lines in between are all streams. And so we start at I-1 as the first lake, I appropriately named, I know. And it's this drawing is representative of it going down the watershed. But it shows how this water is traveling downstream. And then the question here is, are there different bacteria living in the streams compared to the lake? Because these streams are continuously bringing in soil bacteria. And I've seen that firsthand. Like when we are in the streams, like the stream water is usually fairly brown. And then when we collect lake water, the lake water is fairly clear or even looks blue to my eyes. But what's most important here to know about the I-series is that they're all connected. So it's the bacteria and essentially the soil carbon that is, replenished in these different sites, and is then sent further downstream where, again, more soil comes in and gets into the next lake, into the next lake, into the next lake. And so they're trying to identify exactly where these different ones are. Next slide. And so, Kara, in terms of your question of how are the lakes connected, are the small streams between, or do they overflow during certain times of the year? There's definitely a lot of both inlets and outlets of the lakes and the streams that they're essentially leading to each other for the most part in this particular series. So there are small streams, there are some bigger streams. It depends on which lake you're at. And I'll let George answer the rest of the overflow question in the chat. And so this is also from a paper that Dr. Crump has published. And yes, there are different bacteria that live in soil water, that live in headwater stream water, so stream water and lake water. But there is overlap between all of them in different locations. And that's really important because they're trying to identify both the genetics but also the genetic capability of what each of these different bacteria can do. Next slide. Ooh, go back. So the I-series was a set of 11 lakes and streams. And another very 2 full days of field work was spent going to about 36 lakes in one particular area. And so Dr. Crump's lab is looking at how these microbe, fingerprints are changing across the watershed, but on different scales. The I-series was maybe within a few kilometers, but the bacterial survey, which is what this is a picture of this right here, was a set of 36 lakes in the tundra, and those were between 6 to 20 miles away from each other, altogether, that was the range versus a couple kilometers. And so they're essentially taking what they've learned about the I-series lakes and streams and applying it to a much larger scale. But again, still their question is, how are these connected on the landscape, and how does the watershed influence the types of bacteria that will end up further down the stream? Yeah, there's definitely a lot of lakes up here in the Arctic tundra. A lot of water. Next slide. And so this is kind of what a lot of our, field work day-to-day stuff ends up looking like. We collect a lot of different water samples. In the picture on the left, that's me in one of the lakes for the bacteria survey, and I'm wearing chest waders so that I don't get wet. And we rinse all of our bottles 3 times, and then we collect the water, and all these lakes would take us weeks, maybe years to hike in between, and so we actually have a helicopter that takes us everywhere. It's beautiful and amazing, and I have tons of GoPro footage that I can show another time. We also want to collect other data while we're at a particular place. And so on the right-hand side, you have a picture of the lab tech, Johanna. She's taking pH and conductivity of each location. And then we also do bacterial production, which is what Graham, which is the second person in that picture, is doing over there. And then how long does it take to analyze all this water? I'll answer that within the next few slides, but it's definitely over a long period of time because there's many different steps involved of all of this different work. Next slide. So as I said before, water is collected in both the lakes and the streams. So this is a picture of me in one of our streams of the I series. This was after we had this random snow day up here in the Arctic. I know someone had said that it could snow any day of the year, but I didn't believe them until it snowed. And you can see in this kind of picture, where would soil bacteria be coming from, where would stream bacteria be coming from, and where would lake bacteria be found. Next slide. So I've tried to compare a lot of this to a microbial crime scene, and so we got to be able to collect our criminals. And so sometimes, depending on the particular field day, we'll process them in the field, and sometimes we'll stick 20 collected water bottles back, into the helicopter, and we'll fly it into the field station. We'll do it at the field station. And so what we do, or what I'm doing in this particular picture, is I'm holding what's called a caulking gun, which makes filtering significantly easier. We pour sample into this syringe that has a filter at the end of it. And this filter collects all of the different microbes and spews out essentially sterile water on the other side. So there's a whole bunch of, bacteria in that little white filter. Where's the little arrow thingy? Yeah. So at the end of the blue thing in the middle of the picture. Even further, just a little bit. Yeah, right there. So it's called a Sterifex filter. And it's a very small, like 0.2 micron filter, and it captures all those bacteria for us to process, and it's right in there. Whenever I do it, I feel like they're collecting all together and just screaming. So many microbes. Next slide. And so, once we have all these filters, we need to bring them back to the lab. We crack the filters and remove the top of them. We process the filter paper that's on the inside. We also do DNA. Like we have Johanna, which is the lab tech here, and she goes further to also extract DNA from the filters themselves. And then these DNA extractions are sent to Dr. Crump's lab in Oregon. And over there, the DNA is amplified. And so when they do that, they make like millions of different of the same copy of DNA from a particular thing. And then these copies are sent to another place for sequencing. And so all of this happens because, Dr. Crump, he does this to try and quantify the types of bacteria that are found in each of the water samples so that they can represent which, lakes and streams are doing what and which ones and how they're all connected to one another. Thanks, Judy or Janet. I'm glad you like my evil laugh for bacteria and mosquitoes. Next slide. And so you might be wondering, how does knowing what their DNA is help us identify the different microbes? So essentially what we're doing is we're dusting for fingerprints in a specific area, and we're looking to identify the sources of these fingerprints. And we actually barcode all of the different DNA with essentially a label that says stream, lake, or soil. And so when we get the sequence data back, and we've identified exactly which types of microbes there are. You can sort them easily into their soil, lake, and stream categories. They're essentially just tagged to be able to do that, which is very handy when you're trying to study that. Next slide. And so, what has Dr. Crump's lab learned from DNA fingerprinting of these particular microbes? And so what you're looking at in this particular picture is, remember how we talked about the permafrost at the beginning of the presentation? Well, as it thaws, it also, especially the ones that are near lakes, they start to, this is what we're looking at. It's called a thermokarst, but essentially what that is, it sinks further down and it dumps a whole bunch of that soil carbon into the water. And then this, and so these soil microbes, are seeding lake microbes through some of these streams. And what they've also found is that they do find soil bacteria in the lakes, and then slowly the lake bacteria are then equipped with this genetic toolbox to actually convert soil carbon into CO2, which is not good because with the permafrost thawing more and more, it'll keep dumping this carbon into the lake, and the microbes there are able to convert that soil carbon into carbon dioxide, and that'll just continue happening as our climate warms. And we know that they can because by studying the types of specific species of bacteria and where they come from, they've been able to show that these bacteria, the bacteria that came from the soil that are now in the lake, they have that genetic capability to do that. Next slide. And, as great as this discovery all sounds, it's not really a good thing in the eyes of climate change because it'll continuously add to our our positive feedback loop of permafrost thawing. You have more CO2 being added to the system. You have other, heat-trapping gases like methane that are, and both of these are created through microbes, and they're continuing the amount of greenhouse gases that are warming our planet and will be causing future damage to our Earth and to us. But that movement of microbes is also increasing that amount that's being emitted into our planet. Thank you, Dr. Kling. Next slide. And so that's a very short summary of what I've been learning with Dr. Crump and his lab up here. And so to show what I've been doing up here, or what I did before I left for my trip, what I've been doing here, and then what I hope to do after my trip with everything that I've learned. I work with a lot of students. I work with about 3,000 students on a yearly basis, and I really wanted them to feel like they were a part of my expedition. And so before I left, I had each of my students learn a little bit about the project that we were doing, what we were doing with these microbes up here in the Arctic, where was I going, etc. And they each had to, one, ask me a question about what they had heard and draw themselves as a polar scientist. And so I got about 1,500 polar postcards from all of my students, and they also had to draw themselves as a polar scientist because I wanted to see them, I wanted them to see themselves not just as a scientist but also as a polar scientist. So I got over 1,500 questions that I answered. I answered all of them before I left for my trip so that they could actually have their postcards back before the school year ended. My hand still kind of hurts when I think about it. And then what I also did is I had each of my classrooms decorate a flag of polar science for their classroom. And so you can kind of, one of my classes is holding up the flag in the middle on the left, and I have a few students decorating a flag on the right-hand side. And next slide. And for all of the blogs that I've been posting, I have photos of each of the different flags in the different locations that we go to as we learn different things. So a lot of my blogs have their flag attached to the end of it. And so like this was from the day that we went to the I-series in front of the helicopter that brought us to one of the higher lakes with one of the classroom flags. And then I'll also be writing, I'll be writing a polar science curriculum for my K-3 students. There'll be workshops for both teachers and students in the fall and spring. And I'm looking forward to translating a lot of this for them. The next slide. And you know, I come from USC, and our motto is fight on. And so it's fight on for science in this case. And that's all I have. What questions do you guys have? And actually, I can turn on my webcam now, right?
Yeah, go ahead, DJ. Yeah, so there was, I know we have a couple people that had joined just by phone and not actually following along in the chat. I want to ask them very quickly if they have some questions, and your phone might have been muted by me, so *6 to unmute. So Levi, do you have any questions? Because I know you weren't following along on chat, and some things got clarified as we went along.
Hold on one second. Hey, Graham, can I turn on the light? Can I turn on the light?
All right.
Sorry. I needed to make sure I had permission to turn on the light here in the dark room. So.
Okay. All right. So if you have questions, feel free to raise your hand. You can click on the little person up there above everybody, or unmute your microphone and just ask your questions to a small group. So anybody have questions?
Yeah. Can y'all hear me, or?
Yes, we can. Go ahead.
Oh, okay. Are you only looking? You said bacteria. Are you also, by that do you mean bacteria and archaea, or just bacteria?
They are primarily looking at bacteria. Archaea is in that earlier category of microbes, but they're primarily looking at bacteria. Is that Levi?
Yeah, that's me.
Hi, Levi.
Hey, DJ. And then I guess the follow-up one is, I know this is probably too costly, but is anyone looking at isotope ratios for any of the samples you're collecting as well? For
I
carbon
believe
isotope?
so. Yes,
Okay.
I believe that's what the bacterial production is.
Oh, so they're looking at that carbon-12, carbon-13 ratio and just seeing like how much of it is actually going through something that's alive?
I believe they use leucine in this case. I believe they're using amino acids. But I can check and get back to you on that, Levi.
Okay.
Great question, though.
Thanks. I know it's really selfish, but this is going to be like really similar to my thesis.
Or was that Dr. Crump? He might be a better person to ask.
Yeah, can I chime in?
Too. Yes, please.
Yeah.
I don't think stable isotopes are used at Toolik for a lot of different things, but they're not integral to the project that DJ and I are working on. She's talking about using radioisotopes to measure rates of bacterial growth, and that's a different thing. But we do measure bacterial growth rates.
Okay.
Okay. Check.
Okay.
Hi, DJ.
And DJ, I guess your dad was asking a question. Why does it matter that there are different bacteria along the streams? They finally all end up in the Toolik Lake and convert carbon. So I don't know if you want to answer that, or the Dr. Crump or Dr. Kling, but go ahead. You can start, and we'll see what they have to add to it.
Well, because the reason that's important is because they're trying to see exactly who is going to be contributing or converting this carbon into CO2. Because normally lake bacteria don't do that. Lake bacteria, or they don't do it at the level that soil bacteria does. But if there's more soil bacteria that ends up in the lakes and continue, and this may, happen with more permafrost thawing. This will continue to happen, and more of the soil bacteria will end up in bigger lakes like Toolik Lake, and there will also be another source of carbon that will be added to the atmosphere. And I'd love for Dr. Crump and Dr. Kling to help me out with that particular question.
This is Byron. I think it looks like George is typing an answer. Can you restate the question for me?
And he asked, why does it matter if there are different bacteria along the stream? They finally end up in Toolik Lake and convert carbon. Why does that matter?
Why does it matter? Well, different bacteria have different metabolic capabilities, so they can, so they specialize in different types of organic matter. And we found that bacteria in streams, let me see, how did it work? Bacteria in lakes do not grow as well, and they do not process organic matter from streams as well as the bacteria in the streams themselves. So the that difference in ability to handle the food that's available to them is what drives the changes in the bacteria along the landscape, right? So lake bacteria do best on lake organic matter, and stream bacteria do best on stream organic matter. So what matters for carbon cycling is where do they do their work? Are they working in the streams? Are they working in the lakes? And which ones are active? Does that make sense?
And essentially tying that back onto how is this going to impact climate, and what, how is that going to impact us in the end? That's why it's important.
Okay. Kara's asking, DJ, how many people are working on your project? How many different research groups are at Toolik Station right now?
So in my particular group, or at least here in the field, it's been primarily Dr. Crump, Johanna, which is an undergrad research tech that's here as well. And then we have Graham, who is our bacterial production person, and myself. And so that's been primarily the microbe group, but we are in essentially a larger umbrella lab of the dry lab, which also has photochemists. It has, Dr. Kling works a lot with different gases and collecting gases from different areas. It's essentially a giant super group of, carbon cycling and microbes and how all of these interact with one another. But there's about, I believe the number right now is about 80 total researchers that are up here. That includes, its own different groups, but there's bird researchers up here that are looking at migratory patterns and how they're changing with climate, especially with different food availabilities. There's, there's people that are looking at the different vegetation of the tundra, and they have different greenhouse setups with really fancy cameras that can actually see how the different plants will react to warmer temperatures or colder temperatures. So they can they're essentially making climate models and looking at that Arctic response. How is the Arctic going to respond to climate types of questions? I think that's a major theme up here, is how is the Arctic responding, or going to how is the Arctic changing in response to climate, and trying to model it at the best that they can through different means, whether it be microbes, whether it be photochemistry, whether it's birds, bugs, etc. But there's a lot. It's just fascinating to learn about all the different people that are up here. Lunches, dinners, breakfast, they're never boring, because you can just talk to another person and be like, what are you doing today? We have people that collect fish, and they're looking at, both connectivity similar to us. like between the different streams and lakes, and will those populations of fish change, et cetera.
Okay, cool. Yeah. George, you have been typing a lot in the chat box trying to respond to some earlier questions. I don't know if you want to elaborate on what you said, or you feel that you're good there.
Well, I can certainly, if somebody else has questions or if it wasn't clear, they can ask. I can start with trying to answer Martin's last question. What's going to be done with this research? Obviously, we're cataloging what bacteria are where, but is there more than that, he's asking, and are we recommending something more than that we should slow down global warming? I think almost all of the work on climate change has at some level this element to it, saying that we're finding out what the impacts are of climate change, and we know that many of those impacts are not going to be good, or they're going to be difficult to adapt to. So there's very often this idea that we should slow down global warming. But it's more than that, and from the science side, what we want to know is how can we predict what's going to happen in the future. So in order to do that, we have to have a better understanding of how all of these organisms work together. Simply saying that the permafrost will thaw and more carbon will be exposed to bacterial degradation and will make more greenhouse gases, that's fine. That's going to happen. But the real question is how much and how fast. And so a lot of our work is trying to put numbers on the rates of the thawing and the rates of the movement of carbon from land into water, and then the rates that the bacteria are converting it into greenhouse gases. So I hope that helps a little bit.
Looks like it was good. Good answer. Thank you, George. Anybody else have questions in there? Looking around. Let's see. I think we're good there. Anybody else got things that they want to ask? Got a good team here to answer lots of good questions. And I don't know if Levi, since you are not seeing the chat, if you have any questions, feel free to ask them as well.
Well, I guess, do you have, when does the sequencing start?
Well, he has many years worth of sequencing as well, and I can definitely share with you some of the papers that I've gotten as like required reading for my trip up here, because I know actually I might just put you in contact with Dr. Crump as well. I think he'd be a good research for you.
Okay.
Dr. Crump, Levi is a colleague of mine that's in the PhD program at USC for marine environmental biology, and he's focusing on how, remind me, it's how microbes are changing in terms of like ocean water and shipping channels.
So specifically looking at the impact of on microbial biodiversity in port waters, and then like how is that impacted, if at all, by the volumes of shipping between different ports.
Sounds interesting.
Cool. Mm-hmm.
So,
I thought Dr. Crump would like that.
yeah, my guess is
I will put you two in touch.
Oh, okay, cool.
I'll make myself a note right now.
Thanks, DJ.
Yeah, so DJ, I have a question for you, just because we were talking about this last week with a group of teachers. How do you know, this is, it's a little bit more advanced science and everything. So, you know, what's your story, or how do you think you're going to bring this back to the kids so that not only they understand it, you can kind of use the crime scene theme that you've got going on, which makes sense to me, or, just kind of, how do you want to bring this back, and what do you think what's your most important message that they should take away from this science?
Well, essentially, many kids don't really understand what microbes are, and so some of my intro lessons will definitely be like, what are microbes? Actually, give me like 10 seconds. I have something, one of the activities that I wrote up so far that I had my kids do before I left. Let me grab it real quick. It's in the other room. Hold on. Give me like 10 seconds.
Okay. The power of video. She gets to run away and come back. So for those of you that don't see DJ on the webcam, she just left the room and was coming back. There she is.
So I had my students make these as kind of DIY microbes and bacteria.
So
there's
she's showing us the little microbe for those of you.
And so there's a vial on the inside of beads and representing proteins, representing DNA. And so having students understand, what do microbes even look like, even though they're super tiny? And then, so like that one. Trying to avoid so you can see what's on the inside of a bacteria, and then cilia on the outside. So very simple, hands-on, something to touch. And some of the activities that I've thought of so far, microbial biogeography is definitely, Dr. Crumb's bread and butter. And so some type of watershed activity where like there's different beads that start in different places, and having them kind of all see, like adding water to the top and watching them go to different places. So if they're in the soil, if they're in the stream, and do they end up in a giant lake at the bottom? And so just trying to figure out what are the messages that I really want to say. So microbes moving between different parts of watersheds, and then ending up at how does this contribute to climate?
Yeah, I could totally see you having, using different colored beads or something, and having them, kind of building a little model. Peggy is asking about the microbes that you just showed us to do your own. She wants to have directions or a pattern.
Yeah, no, for sure. I can definitely send that to you, Peggy. I love these. I had my kids make them for all the researchers up here before I left.
That's very cool.
Yeah. We also usually put pipe cleaners on. So it's hand sanitizer. It's part of a microbial unit that I've done with students. So it's usually beads and, for proteins, pipe cleaner for DNA, and then hand sanitizer is the cytoplasm. And so if you ever get sick of it, you can always use the hand sanitizer as a way to, kill some of the bad germs on your hands.
Nice. I love it. All right. Any other questions going on there? I think we've done pretty good. There's a really interesting presentation, and I'm going to switch screens on everybody here. So DJ, you just disappeared. Sorry about that.
Okay.
In your webcam. But we want to say thank you to everybody that was able to join. There was a question about archiving, and we will archive this, and we'll put it on the website and with a link, and then we'll send it out to everybody that registered as well. So it was very interesting. New science for me. So thanks. You guys did a great job, and it was really nice to have Dr. Byron and Dr. Crump, and Dr. Kling on board as well. Thank you for responding to all those questions.
Anytime.
All right.
Pleasure.
And we're just going to sit here for a moment, so we'll let everybody answer the poll if you get a chance. And then feel free to sign off. And DJ, if you want to hang up your phone and just do voice over IP, that's fine probably now. And I'm going to also stop the recording.